Our next, now we're going really down the line of thinking about this whole human and habitats, still talking about the challenges and how do we understand the challenges for interstellar flight. We'll be able to apply some of those ideas as we go through. Our next speaker is Dr. Pam Contag, who is going to come up from the staging area. Dr. Contag is what we call a serial entrepreneur. Right now, she's currently the CEO of Signet Biofuels. She's founded 4 venture startup companies, but her background comes out of the field of immunology. She has her PhD in immunology. So she's going to talk with us a little bit about those things that need to go with us, but that we don't necessarily think need to go with us. And that's the microbiome and humans in the loop, her perspective as a microbiologist.
Thanks to ISC and to May for inviting me on this particular journey. I don't plan to get on a starship anytime soon, but I have learned a lot. And one thing I want to point out, and thanks to all the speakers, I have learned so much over the last week of spending dinner times with them and everything. So I understand that Ronki wants to take her red wine, made from a microbe, by the way. I heard earlier that May wants to take her cat. And what I want to say is they have 2 things in common, and that's they carry hitchhikers with them in the form of microbes. So I want to talk to you about, and I noticed, Ronki, in your picture of your human, like this, I didn't see the gut teeming with microbes. And microbes actually would be affected by space conditions also. So that's one more thing to add to the picture. So how do we innovate and integrate to create regenerating closed-loop systems for human survival in unfriendly environments? And one of the reasons I study microbiology is because microbes have the amazing ability to adapt to new environments. And they do it in a myriad of different ways. But when we think about humans, we have some basics of short-term survival. And this is what you use when you go out on your 2-week camping trip into the desert. 3 minutes without air, 3 hours without shelter, 3 days without water, 3 weeks without food. Well, you know, that is not going to work on a long-term space travel flight, or even, you know, as Mark pointed out, even in Earth as a spaceship. The fact is that to survive, we need environmental diversity, we need an intact food chain, and we need health and wellness. And if we don't have those 3 things, we can't get to, you know, Catherine's wonderful point about we need a mechanism of politics and decision making that's, you know, ethical. And so the reason I study microbes is because microbes play a role in each of the 3 things I mentioned. Environmental diversity and sustainability, an abundant food chain, and health and wellness. And I started studying microbes, let me see, the first extremophiles were found over 40 years ago, and it was 10 years later when I first took my microbiology course. And one of the things that happened in that course is that I had to learn the names, genus and species of many, many organisms. Not as many as we know about today, but many. And at the end of the course, this professor, who I remember her name was Mrs. Lennon. She didn't have a PhD. She was teaching a small course in bacteriology. And memorizing names wasn't my strong point. I learned that I actually had to read it before it got memorized. So I wasn't so great at that. And so the last test was, okay, invent a microbe using the chemistries that you can think of or imagine that's carbon-based. And the second part was, okay, invent a microbe with any chemistries, real or imagined, that's not carbon-based. So we all handed our papers in, and the people that used carbon-based got a big A. And she said, okay, listen, you can find all of these microbes, no matter what chemistry you used here on Earth. Start looking. And the people that didn't use a carbon-based microbe, she gave them a big A and said, you might find these on Earth, but most likely, if you want to find these, you're going to have to go out into space. And so what that taught me was there's incredible diversity, and I was inspired to go look and see what that was all about. So one of the things I learned over the past, I don't know, I hate to admit, 25 years, is that every organism, plants, animals, have a unique microbiome. And I put the cat in there for May Because one of the things that's important is that we actually share microbiomes with the soil, with our pets, with our children, with plants. And the ability for those microbes to adapt are actually influenced by the fact that microbes can talk to each other. And I don't know if it's appropriate to discuss here in Parliament, but microbes actually have sex. And so what happens is either small mutations can occur or large chunks of genome can be moved back and forth. And that's one of their amazing abilities to adapt. So the thing that I study, and I'm going to go through the next slides very fast because in the end, the whole point of this panel is to invite collaboration. I study the carbon cycle. So in many ways, climate change caused by industrialization is disrupting our carbon cycle. And so if you want to simplify that, I simplify it by simply saying microbes make the world go round. They're in every aspect of what we do. Our ecosystems require microbes for stability and thus for human survival. And I just want to remind you that those are the 2 points, leading to what's actually a society. So when I was talking to my friends here, I think everybody realized it's not efficacious to eradicate all microbes or even to target all pathogens, because many pathogens actually are also the good guys. They're sort of superheroes by day, pathogens by night. And so many questions remain. So these are the questions around which I think collaboration is really valuable. The role of vaccination. Can we sync our microbiomes and actually eradicate the pathogens from our microbiome? What's our response when new pathogens are encountered? I think we need things like cell-based therapies and ultra-fast drug development. So in essence, in order to get there, we need to understand carbon cycling. We need to understand recycling and sequestration of carbon. And that relates to everything we find dear in our life today, which is wine, energy, and pure water. How do we measure microbiomes in new environments? That's one thing I spend a lot of time on biosensors. And can we build a sustaining closed-loop carbon and energy system here on Earth for the end-to-end trials that we know we need to know before we go into space? And what are the nutritional requirements for humans and animals? And how can the microbiome play a role? In fact, if you're actually going to be all vegetarian in space, you're going to have a microbiome that can handle that. And of course, what are the mechanisms and risks for cross-species diseases? So I'm hoping together we can develop a vision for the grand challenge of building closed-loop systems on a huge scale. And we have to be able to understand ourselves and our environment at every scale. So invite you to collaboration. And I'm from, with May, 100-Year Starship, VTT MSI. We do single-cell analysis. My companies now do microbial diversity in the carbon cycle for the purpose of biofuels currently, and Stanford University, where we study human disease. And thanks again to everybody, and thanks to my collaborators back in the U.S.
Thank you very much. Thank you very much, Dr. Contag.